And this is an area where OTR lab is directly supporting that feedback, and OTR scientists have published a number of papers on MAM, and that science being done in the laboratory is actually directly informing that feedback we can give to sponsors as they develop their applications, both for product development and eventually for QC applications.
Thank you for responding to that question. The next few questions will go to Dr. Patel. And here's the first question. Does the agency expect a leachable study from the manufacturing components, VIZ, filter, tubings, gaskets, etc., or a final leachable on the drug product would suffice with a summary of extractable data on the manufacturing components?
Hello, this is Kshitij Patkar. It's a very good question. We do expect leachable study from manufacturing components if you carried out extractable studies on the manufacturing components and found extractables above analytical evaluation threshold. However, if you chose to do a final leachable study on the drug product, we expect you to evaluate those extractables that you found about AET in the process extractable studies. So you have an option of doing separate study, or you can do it on the final drug product, but then you have to evaluate those extractables that you found from manufacturing equipment about AET. So your analytical method should be capable of picking those up in the final drug product.
Thank you for responding to that question, Dr. Patkar, and we were hearing you a little muffled at the very end. You're able to readjust your mic, and we'll have another question to try out and see if we can hear you very well. And here's the question. What kind of quantitative data is expected for extractables studies?
We expect that you provide quantitation of each extractable component that was found above analytical evaluation threshold individually. So for example, a lot of times we see a data for total NVR, but when you see the total NVR, they are above analytical evaluation threshold, then we expect you to individually identify the compounds in those total NVRs. So that's the kind of quantitative data we expect you to provide.
Thank you, Dr. Patkar, for responding to that question. We have one more question for Dr. Patkar, and here's the question. How do we determine the right conditions for extractables studies for manufacturing equipment?
You should look at the drug product formulation and your process parameters, as I have presented in my talk, that the extractable conditions should exaggerate your manufacturing process conditions. So for example, you have 20% ethanol and neutral pH and filtration at 8 degrees, then you can have 50% organic solvent at a higher temperature. And also the contact time. If your filtration goes on for 24 hours, then your extractable study should be at least 24 hours or more.
Thank you for responding to that question. The next group of questions will be directed to Dr. Gou, and here's the first question. Could in vitro lab science methods be used instead of in vivo equivalence / clinical endpoint studies to compare RLD and generics? If so, when?
Yes, this is a very good question. I would say this is happening depends on the complexity of the drug products and the delivery release mechanism. Some of the drug products already have some guidance document. For example, for nasal suspension products, there are several product-specific guidance already written in for the alternate MDRS method as a replacement of clinical endpoint study. So it could be much more complicated for some other dosage form. For example, for MDI product, the small testing may be involved, such including MDRS, biorelevant mouth throat models, spray velocity measurement, and dissolution. I would say it is happening right now, and we are working on to develop more methods to make it to apply for more dosage forms.
Thank you for responding to that question. The next question is also for Dr. Gou. For solid orals, does your lab employ biorelevant dissolution methods to evaluate in vivo and in vitro correlation between the RLD and ANDA is this under consideration?
Yes, we have a handful of ongoing projects related to the oral solid dosage forms on the biorelevant dissolution method development and evaluation. And overall, for most all the type of dosage forms, development of in vivo predictable in vitro method to replace the expensive clinical studies is a major area for OTR labs. Thank you.
Thank you for responding to that question. We have one more question for Dr. Gou. What is your opinion to use MDRS to test API and DPI products?
So MDRS already shows potential to measure API particle size in dry powder inhalers. An early study already performed by University of Bath Research Group in UK, and the result was presented last year in RDD meetings. So there's for sure for DPIs, there's much more challenge and the complexity in nasal spray. First of all, the API particle size is normally smaller than nasal spray. Second, the API and excipient agglomerate is a big challenge in DPI product. However, MDRS already showed the potential to perform this type of testing, and it's just a little bit more challenge than in nasal spray. Thank you.
Thank you for responding to that question. We're going back to Dr. Patkar. We did have a few more questions come in. And here's the first question. Do you recommend following USP 1665 risk assessment guidelines for process contact materials? According to USP 1665, not all the components for all formulations not required for full chemical characterization, most often NVR, delta, pH, would be sufficient. Would you concur with this approach?
Hello, hopefully this time I sound better. That's a good question. USP 1665 is still not official. I think you can take a look at it. It's a good guidance. However, like I said, you should do your own risk assessment based on drug product formulation and process parameters. And regarding not all components require chemical characterization. So if you did total NVR, and if you found that total NVR are above analytical evaluation threshold, then you're expected to then further identify individual compounds from the total NVR and then quantitate them. So you may have only few of the compounds from the total NVR that may be above AET, and then they may require further assessment, either tox assessment or leachable studies, etc. So definitely you are expected to do the NVR and then follow that up. But we also expect you to carry out semi-volatile and volatile component assessment.
Thank you for responding to that question. The next question for Dr. Patkar. Can you provide any guidance in regards to selecting a model solvent? Like, would it be based on polarity or some other factor?
Model solvent selection should be similar to what you would do for container closure system extractable leachable studies. Those you can follow those guidances in USB 1663, 64, but please make sure that your solvent is relevant to the drug product formulation. And yes, you can use polarity as a basis for selecting the solvent. So if your drug product formulation is neutral with some organic component, then definitely match that organic component in terms of polarity and in the selection of the solvent.
Thank you for responding to that question. We have one last question for Dr. Patkar, and here it is. Should a gasket included in the train that is not likely to contact the flow but has a slight potential for contact be included in an extractable assessment? Also, can certain food certifications be used to support the gasket's use? If the food grade certification is not adequate, how do you determine the actual exposure?
You should definitely take a look at your process, and if the contact time of the bulk with the gasket is minimal, you can include that in your risk assessment and provide justification accordingly for not including that in your extractable leachable assessment. I have seen applicants actually use the whole tubing equipment that includes everything, tubing connectors, as well as if there is any filter attached to the tubing, and carry out extractable studies on the whole capsule along with the tubing. You could do that. If you choose not to do the gasket or remove or not include gasket in your studies, just provide a justification for that. And food grade certification should be provided for all equipment. That is a GMP requirement, irrespective of whether you provide extractable or leachable studies or not. Food grade certification is required for all polymeric equipment surfaces.
Thank you for responding to that question. The next group of questions will be directed to Dr. Chou. Here's the first question. If there is no extractable found above AET in a simulated extraction study, do we still need to do a leachable study, or can that be waived?
Is addressed to both as a product question and a process question. I am going to attempt to answer this question first, and Kris can back me up. So to begin with, the question says, if there's no extractable found above AET in simulated extraction study, that scenario doesn't usually happen in an extractable study because usually we found something if the condition is harsh enough. And usually the level is, well, for a good container closure system, the level of extractable found would be low. But in a simulated extraction study, even when the AET, when the level of extractable is found to be below AET, sometimes the reviewer will still request a leachable study for the following reason. First of all, there could be components, migratory components that leach into the product over time. And second, there could be process-related extractable that come to interact with low levels of extractable with container closure system, and they could grow over time. And it's not something we can foresee. A lot of the assessment we do is risk-based, and a lot of the times we based on our previous understanding of products that we have reviewed before. So the short answer is yes, sometimes we do require leachable studies even if the level of extractable found is low. And I don't know if Kris wants to add to my answer.
Hi, Grace. Just one extra point I would like to add. If just because you did not find compounds about AET, that doesn't mean that they were not there, because it depends on your extraction conditions. If your drug product formulation contains organic solvent, but your extraction conditions did not use organic solvents and use just water or acid, then and then you said that there were no AETs, there are no compounds about AET, then most likely the reviewers will ask you to repeat the studies with appropriate extractable conditions and solvents. So yes, I agree with Grace that just because you did not find compounds about AET doesn't mean that you will not be asked to perform leachable studies. It all depends on if your extractable study protocol was adequate, if your analytical methods were sensitive enough. So just be aware of that.
Thank you for responding to that question. We'll go back again to Dr. Chou for another question. Are there any plans of issuing a guidance document for control of EL and drug products?
We have a dedicated group working on the issue, and hopefully in the future we'll have a very clearly defined guidance document on all aspects of extractable and leachable for drug products coming out.
Thank you for responding to that question. We have one last question for Dr. Chou, and here's the question. Is the leachable study required for all 3 exhibit batches or 1 batch?
Leachable studies at the end of the shelf life, usually we ask for all 3 batches to be tested.
Thank you for responding to that question. The next group of few questions will go to Dr. Mueller, and here's the first question. To assess the CSS for EL for parenteral injectable drugs with APIs with genotoxic and sensitization nature, what SCT level should be used for calculating AET? 1.5 micrograms per day or 5 micrograms per day, or since the drug product itself is genotoxic in nature, can we consider product's maximum daily dosage, or MDD, as SCT for leachable assessment of the drug product during the stability?
Great, thank you for this question. For injectables, we recommend using an SCT regardless of the nature of the product of 1.5 micrograms per day. Everything that exceeds 1.5 micrograms per day as extractable or leachable should be characterized and identified. Depending on the context of use of the proposed product, then at least a qualification threshold of 5 micrograms per day should be applied to inform general toxicity. And then in terms of gene tox assessment, borrowing principles from ICH-M7, if the product is indicated for less than lifetime use, you may apply those thresholds appropriately on the proposed duration of use to inform genotoxicity assessment. And certainly if you're dealing with a cancer product for injection, you may leverage the carcinogenicity or the mutagenicity of your API in your tox justification. However, this will likely be a review issue. And during the assessment, the safety assessment, we in OJD certainly align our approaches with that of the R&D division. I hope that answers your question. Thank you.
Thank you for responding to that question. We have another question for Dr. Mueller. And here's the question. Currently, there is no guidance from FDA or USP on safety concern thresholds. Can you please describe FDA's expectations for SCT levels for injectable ANDA products? Are there different SCT expectations from FDA for liquid versus lyo or chronic versus acute use of the product?
Great, thank you for that question as well. As I just reiterated, typically we would recommend characterization and identification of everything that exceeds 1.5 micrograms per day. If you're dealing with a chronic use product, certainly genotoxicity of any leachable that exceeds this threshold should be identified. For acute use products, again, you may borrow the principles of ICH-M7. Let's say your product is indicated for use of a month or less, then for gene tox assessment, you could apply 120 micrograms per day. However, I'd like to remember that 5 micrograms per day is what we use as a qualification threshold for any toxic endpoints that are non-carcinogenicity. In terms of liquid versus lyophilized, I think we would apply the same thresholds. I hope that answers your question. Thank you.
Thank you for responding to that question. We have one more question for Dr. Mueller, and it'll be our last question for this morning's session. And here's the question. How does SCT need to be selected for a liquid injectable for the EL study?
Thank you. And I feel like these questions are all very, very similar. So yes, SCT to identify, characterize anything that exceeds your AET, I would use 1.5 micrograms per day. And of course, the appropriate maximum daily dose to set your AET. Anything that exceeds the AET should be characterized, identified, and then using 5 micrograms per day for general tox assessment, and then the appropriate limit based on the treatment duration for gene tox assessment. Thank you.
Thank you for responding to that question. This concludes our Q&A panel for this session. We just have a couple of housekeeping reminders. In addition to the speaker evaluation, we have our final evaluation, which will be available today. And remember, this is the final survey mechanism by which you will receive the certificate of attendance, which supports CEs for RAP, SOCRA, SQA, and ACRP. Also remember, the evaluation and certificate are only available for 2 weeks, and that's May the 14th. So please be on the alert for our thank you email, which will be sent out in approximately the next 24 hours. And as a reminder, all sessions are being recorded. You'll soon be able to access the recordings on YouTube from SBIA's webpage at fda.gov/CDER-SBIA. Also remember that in addition to the certificate of attendance, which supports the CEs for SOCRA, RAPS, SQA, and ACRP, this conference is offering a total of 13.5 hours for CME, CNE, and CPEs for physicians, nurses, and pharmacists. These CEs are available to all attendees who identified as a healthcare professional during registration.